Okay. Okay, thank you for the opportunity to talk about forestries. It's not the plants that you heard about that form crops or orchards that are spoiled with water irrigation. We are studying forestries, and we're trying to understand their ability to survive in nature when it's very dry and hot. So when we have trees in the forest, they have three options when drought comes. Is it to habitat tracking, like to move to another habitat that is much more convenient for them, or go extinct, which happens a lot, actually daily around the globe. And we study the third option, which is adaptation. We are trying to understand how forest trees are adapted to drought conditions. So, as you know, when plants are dried, they have less water availability, and when water is reduced in the soil, they don't have irrigation again. So they actually trying to control the water loss by closing stomata. So stomata conductance is reduced. And then when stomata are closed, we also get less carbon assimilation, which is not that good for the plants because it cannot grow and invest in energy. And then when the demand for carbon increases, the storage, when demand increases, the storage is reduced, and then we get a carbon starvation, which is actually a very bad situation for the plant, for the tree. In parallel, when we have water availability is low and stomata conductance is not that reduced, we get water potential reduced in the stems and in the leaves, and then we can get hydraulic failure. And most of the trees that die from drought in nature, they die from hydraulic failure. I'm not going to get into this mechanism, but they are also connected, these two processes, and trees are dying. But some trees can survive as you see in this picture, some trees can survive and actually grow nicely in these dry conditions. So when we want to understand how trees are doing well in drought conditions, we need to get the trees into the lab or to the greenhouse and into the experimental area. But it's very hard to do that in forest trees because they are big, and also another thing that we need to remember, that when we look at the forest when we look in the trees, we know that they have very high variation, genetic variation. Again, it's not like crop or orchard trees, which are identical genetically. They are very different. So if I want to understand how this specific tree survived and didn't, I need to take these trees into the lab. So what we do, it can be done in any species, but we successfully rooted the branches of these trees, and actually we took copies of this genetic background. Like if we take branches and we root them like we do in orchards, or we can take the actual genetics and try to understand how does it grow in and responds to drought. So together with Professor Yassirayov and Einat Sadot, they developed a rooting protocol, and we took roots from the trees that survive in the forest, and we brought them in several copies into the LeZion metric system. So here you see the first, not the prototype, but the first version of the LeZion metric system, and we worked together with Menachem in the Faculty of Agriculture, and Hagar was her master. After that, she did Ph.D. in Weizmann Institute, and now she works there. But her master included this experiment, and she tried to understand how pine trees respond to drought. And it was very precise measurements, And as you see in this graph, we measure the response to drought along 50 days, and then we irrigated them for recovery, and we measure also the response in the recovery phase. So we started this experiment by taking samples, like a few needles from each tree, every 2 days. And we immediately froze them in liquid nitrogen and kept them in -80 until the end of the experiment. Now, at the end of the experiment, we did this analysis. The system was still the old version, so today is much easier to do the analysis and getting very nice graphs. And then we decided to use the stomatal conductance as a proxy for drought response. And we follow the stomatal conductance in the wild type, which is the gray. The control, not wild type. They are all the same genotype. And we follow the control plants, which were irrigated along the experiment. And we compare them to those that are in orange here, which were imposed to drought stress. And then we decided we have needles from every 2 days along the experiment. And then we decided to focus on 6 stages. We call them D1, pre-response. I have the names in the other figure, but we decided to take 6 stages, D1, D2, which is before we see any response by means of stomatal conductance, because by eye we don't see any effect on the plant. It doesn't show along the experiments. After even 50 days of drought, it didn't show any wilting. So we have to count on the lysimetric system to tell us how the plant responds. So we took the D1 pre-response, and then D2 when we see initial stomatal conductance reduction, and then at the end of the drought experiment when the plant is very dry, and a few hours after irrigation, and during recovery, and then full recovery. And we took these needles, we extracted RNA, and tried to understand the molecular response of these trees to drought stress. As you see here, we got very nice results. If we look at the stomatal conductance response in here, with the 6 stages we measure, we see already before we see any conductance, any stomatal conductance reduction, we see already some genes are upregulated and downregulated. And then following the experiment, we see expression, upregulation and downregulation, and that was actually a very, comprehensive experiment showing pine response to drought molecularly. It was the first publication in this issue, so it was very highly cited paper. We also could take all these genes that were behaving the same, like we did some cluster analysis, as you've seen this here, in here. So when we measure the drought-related response and the recovery-related response genes, so we saw some downregulated genes and upregulated genes, and they have some patterns. We know that these genes are sometimes are connected to the same processes. And we took it one step in ahead and tried to understand what genes are connected to a stomatal conductance response. So based on the lysimetric system, we know exactly what is the stomatal conductance along the experiment in the plants that were dried. And we follow this we saw the same genes as you see in these tables. In this table, showing the same response of downregulation and upregulation, and we saw like some of them are connected to osmotin-like protein, to a metin-like protein, which was very expressively highly responsive gene, ubiquitin, and some transcription factors, gibberellin, beta, deoxygenase, and aquaporin. Of course, it was very central gene in this response. Sucrose synthase, which we also know that it cleaves sucrose to fructose and glucose, which are very important for osmoregulation of plants when they are getting dry. So the same needles, we could also send them, this is actually the same data set of RNA-seq, where we also could analyze the response by means of stomatal conductance in response to a hormone response to stomatal conductance reduction. And we saw that ABA was not, where is the ABA in this figure? ABA was not dramatically responding in the first stage of stomatal reduction. And when we send these needles to some company in Canada that measures hormones, it was like 10 years ago. Now we have it in Volcani, all these measurements of hormones, but we didn't have that back then. We could see that pine trees, as you see in here, we have the control plant in the first stage before we see any response. And this is the third stage when we see the dramatic response to drought, the control, which is irrigated, and the plant that was dry, and then the after recovery. And it was very surprising for us to see that most of the ABA is linked to ABA glucose ester, which is a standby version of ABA, and it only needs an enzyme that cleaves this glucose ester to make the ABA active. So that's why we didn't see an ABA response in the first stages, but only later. And as you see here, the ABA glucose ester is reduced in the plants that are dried, and the ABA active form is increases in these plants. So it was very nice to see that, because we had these samples that we took along the experiment, and we knew exactly how the plant responds in each state based on the LeZion system. And another more later experiment we did on okay, it will be very fast, on genotypes. We're trying to understand the genotypes in nature, how they respond to drought, and we have many species in nature, and some of them are planted, and we know that Pinus halepensis, which is Aleppo pine, Oren Yerushalayim, is very closely related to Brutia pine, Oren Brutia. And in nature, we found that they are making hybrids. And we wanted to understand what the value of these hybrids by means of response to drought. So we, okay, רגע, אני צריכה להעביר את זה. זה ככה. So Naomi Hominer was a Ph.D. student in my lab, and she studied this hybrid in nature along the climatic gradient of Israel. And she found that they are very much more. They have a higher, or not higher, but bigger tree trunk, like they have stand diameter. Thank you. The stand diameter was much higher in those hybrids than the other trees in the plot, and we didn't understand why they have this higher trunk. So she took some seeds of the hybrids and the two separate parents, and she grew them and followed the growth parameters, and she found that the hybrid gets parameters that are similar to Pinus halepensis, which is much more resistant to drought than Pinus brutia, but some parameters are related to Pinus brutia, which allows it to make more photosynthesis and grow a nice root system. But we still didn't have answers for that question, so we grew these plants in the LeZion system. And as you see in here, they are very big, and we follow this response along a very long time. I don't remember exactly, but it was several weeks at least. And we found some very interesting results, and we could explain the response of this hybrid, because as Karen showed at her presentation, that this graph shows us when exactly plant closes stomata. This is called the tetacrete point, and we see that the stomata conductance reduces. It means that this hybrid senses the drought earlier than the other parents. So the Pinus halepensis, we know that it senses droughts early before Butia because it's much more resistant to drought, but the hybrid senses the drought much earlier, and it makes it more resistant to drought. And we could explain by all our results, and of course this Lysimatus system, we could explain the resistance of these hybrids in nature. They are much more resistant to drought than the either parents. So, and I also want to thank Gilor about the help with the transcriptomic analysis before when I'm sorry, the experiment that we did before, I showed before. It was very interesting because we found some new findings of transposable element activation, which was never new before. And I just want to emphasize that the Lysimetric system makes it much more accurate. Everything is much more accurate, and we can say exactly what we see at what stage. And in forestry, it was not something that people did before. So it was very, very good paper to publish, and we are happy about it.